Hi, my name is Jennifer Condon. I'm an associate professor here at Wayne State University in the Department of Obstetrics and Gynecology and Physiology. My lab is housed at the CS Mott Centre, and the work in my lab is largely focused on trying to determine what are the molecular events that allow for the maintenance of an appropriate gestational length. We hope in defining the molecular mechanisms that allow for the maintenance of uterine quiescence for 9 months of pregnancy in the human, and for example 19 days in the pregnant mouse, that we may gain insight into why over 10% of pregnant women in the US end up undergoing premature labour and preterm birth. Now we know that being born premature has devastating consequences on both the mom and baby. It also costs the US economy about $10 billion each year. And here in Detroit, preterm birth rates are phenomenally high at 18%, and that's why we chose to work here in Detroit. Now, many groups are working on the potential triggers that allow for the onset of preterm labor. However, our lab is taking a different approach. We're asking, why do we not see preterm labor more often? Why is it only 10%? What are the molecular mechanisms that are maintaining the pregnant uterus in a quiescent state for 9 months of pregnancy? And what are these conserved factors between the pregnant mouse and human that allow for the maintenance of the uterus in a relatively non-contractile state from implantation to term? So what we suggest is that preterm birth may actually be as a result of a loss of function in the ability to maintain uterine quiescence. And our analysis suggests that during pregnancy, from implantation to term, the pregnant uterus is continuously monitoring for, adapting to, and overcoming a distinct series of gestational stresses in order to maintain uterine quiescence. However, an inability to accommodate these stresses actually leads to a surge in a precocious inflammatory response, which ultimately leads to increased uterine contractility and the onset of preterm labor. However, what we have found is that these normal gestational stresses that every pregnant uterus experiences across gestation actually act to precondition the pregnant uterus for subsequent gestational stresses, allowing the uterus to maintain quiescence effectively. And we have also found that this process is absolutely reliant upon activation of the uterine unfolded protein response. And we have demonstrated in our pregnant mouse models that an interruption in the unfolded protein response or an inability to host an appropriate unfolded protein response puts that mouse at an incredibly increased risk for preterm birth. We've also demonstrated in our human cohorts that there are proteins being secreted from the pregnant uterus into the circulation that are associated with the unfolded protein response that can clearly identify whether that woman is at an increased risk for preterm birth. What we have found is that these circulating proteins actually act to modify inflammatory cell differentiation and limit local and systemic inflammatory responses, thereby maintaining uterine quiescence. So that's really in a nutshell what my work is about, but I'd like to talk to you a little bit about the Mott Centre also. So being a part of the Mott Centre really affords us as scientists a unique opportunity and access to a wide range of expertise focused solely on reproductive science, which is quite unusual. Our centre offers an infrastructure that consists of reproductive endocrinologists, reproductive immunologists. There's also research focused on environmental, teratogenic, and epigenetic aspects. We have embryonic stem cell and placentation expertise. We have developmental biologists whose emphasis is mainly on ovarian and testicular development, and we have uterine biologists like myself. I'm also the head of the reproductive sciences graduate program here at Wayne State, and that is also housed here at the CS Mott Centre. And we attract an incredible group of undergraduates, master's students, and PhD students to that program. And we also offer several reproductive courses, from basic principles of reproductive biology to translational topics in stem cell biology. So all of these aspects of the Mott Centre, including our in-house sequencing core, our microscope cores, imaging core, and our soon-to-arrive transgenic core, and also our central location within the Wayne State community, actually allows for the Mott Centre to be an ideal working environment and actually helps our research go in the direction that it should. Thank you.